ऐप सीरीज देख रहे हैं।
Good afternoon, Abir Dey, sir.
Hello, sir. Good afternoon, sir.
Very good afternoon.
Yes, sir.
Just we will wait for 2-3 minutes.
Okay, sir. No problem, sir. I am here only.
Suman Kumar. Suman, are you there?
Yes, sir. Yes, sir.
Okay. Okay. Good afternoon.
Let's start, sir.
Yes, good afternoon. I would like to welcome today's our expert, Dr. Abir Dey, sir. On behalf of the course director, I would like to welcome him for this online training program for the GRF, SRF, and ARF students. Abir Dey, sir, he has completed his MSc and PhD degree from the ICAR IARI New Delhi in 2000, MSc in 2011 and PhD in 2017. During his MSc,
Sir, not needed. Sir, let us straightly go to the class.
Sir, not need all these things, sir. Only, sir.
yes.
Already it's
But, sir, with the small welcome of sir, we start the class, sir.
Okay, no
Yes,
problem.
sir, yes.
Sir, please proceed, sir.
With your some kind
Okay,
of
sir. Thank you, sir.
it's a start of.
Thank you, sir. So, okay, I will just go straight to the class actually.
Yes, sir, please.
Yeah. So, this is the portion we'll be dealing with today. Okay. So today will be yesterday means in the previous class what we discussed actually we discussed about different soil testing methodology for their means different elements. Another one topic actually was interested to me by your by the course coordinator, respected course coordinators, that is methodologies about soil organic matter study. Now we know in soil so as we know, soil organic matter is very important. Means it can we yesterday also we are telling it is the heart of all the soil health. So we can actually know soil organic matter. We can have some of that means some of its estimation from molecular carbon. But if we want to like study the organic matter itself, then first we have to extract the soil organic matter. Now, what is soil organic matter? Soil organic matter composes of different kind of compounds: humic acids, fulvic acids, means humus, means mostly first category is humus, another one is non-humus. Now, humus means the portion of soil organic matter which has undergone some of the chemical changes, some degradation, and it is an intermediary product of the soil organic matter decomposition process. Means if means for that first we need to know the life cycle of any organic matter. Say some plant litter or some animal litter falls into soil, so that is the primary point of that. Then it is having different compounds, some protein, some cellulose, some hemicellulose, some lignin, etc, etc. So then it go decomposition by soil microbes. What is the final point of that process? Obviously, ultimately the final point will be formation of carbon dioxide and water, H2 and CO2. Now, in that process, this humus is an intermediary product, and it is a heavily complex chain structure which cannot be further decomposed by those microbes. So that is what we mean by humus. Now, based on its complexity, its reaction time, and its solubility in different reagents, we have three fractions of humus: humin, humic acid, and fulvic acid. And rest of that, rest of the organic matter is known as like non-humic substances. Now, non-humic substances is mostly the substances that we know the structure of, means which is having a particular structure. Means if I say it's a protein, then the structure is same, that the structure will be same in India, same in Australia, same in any place. Okay. But in case of humic substances, it may be fulvic acid, humic acid, or humin. It's a very complex structure. It's a chain-like structure, and with several interlinking bonds. So it is not having a specific structure. So all those complex molecules, we term them as humic substances or humic acid. Okay. Obviously, now this humus is three types. I mean, it can be divided into three portions: humin, humic acid, and fulvic acid. And this is also based on their solubility and their complexity of structure. Humin is most complex, most interlinked structure is present. Then somewhere less is humic acids, and then the most soluble portion, most easily extractable portion of soil organic this soil humus is fulvic acid. Okay. So obviously, if you have to study that organic matter, we need to first extract it, and this is the extraction scheme of soil organic matter actually. So this humus, as I told you, humus are polyelectrodites, and their complex structure, they remain insoluble in water, and this is having a lot of charges developed there, a lot of negative charges. So generally, their negative charges are neutralized by divalent ions, polyvalent cations, or by hydrogen ions in case of humic acid. So this will make actually a globular structure, a very complex structure in the soil. So that cannot be extracted. It is also having some complexes with clay minerals. So that cannot be extracted. So what do we have to do? We have to make and also we have to make the solution, the extraction medium, alkaline. So when we do this, so generally this organic matter or humus, uh, they are hydrophobic in nature, means they do not uh, means they are not hydrophilic, they are hydrophobic. Phobia means fear, means they do not like to bind with water. So that is why when in acidic structure with charges, they means when those charges are satisfied with your polyvalent cations, they will complex, means they will make some globules, they will coil on itself and make complex structure. So that cannot be extracted. So that's why what do? We means we introduce some extraction agents which are mainly lithium, sodium, or potassium containing reagents, which can actually replace those polyvalent cations. So and also we make them make the solution alkaline, and in that alkaline solution we also introduce the extractants which make this a dilution, means concentrated solution, diluted, means 1:10, 1:20 we can take. And in that alkaline solution and also in high dilution, and when we are supplying with sodium containing extractants, so what it will do, it this globular structure, it will form linear structure, because then there will be unsatisfied negative charges present on that humic acid or fulvic acid particles. So obviously if those charges will be there, so they will be repelling each other, right? And when we introduce sodium actually comes with a very large size with 6 water molecules in itself. So then again, two organic means two humic acid chain structure, they can't be interlinked together, they can't be so much closer to each other. So then it becomes soluble in high pH solution containing some monovalent ions like sodium. Okay, so in alkaline condition and high dilution, these functional groups on humans become negatively charged and repel each other and forming a thread-like structure, which can be extracted. Yes. So that is what we do. We first take some soil, we then reflux with methanol or benzene just to remove. Now, after that, there will be some of the organic matter will be solubilized. So those will be non-humic substances. So after that, we remove, means we do centrifugation and we remove those non-humic substances, take the rest of the material. So that is the residue, and that is basically humic substances, means humus. Now then now that humus we extract with alkali. That just what I was saying then. So that humus we treat with alkali, and in this case we use sodium hydroxide along with sodium pyrophosphate. Okay, we have that procedure. So then after some times of shaking, then one will be insoluble, that will be the humin part, and then other one is soluble. So that means humin is non-soluble in both in alkali and acid. And in alkali, what is the soluble portion? The soluble portion is humic acid and fulvic acid. Then we take that soluble portion and again we treat it with acid, maybe H2SO4, maybe HCl, and bring that acid pH of that solution to like pH 1 to pH 2. Now this humic acid is not, it is soluble in alkaline condition, but obviously not soluble in the acidic condition. Okay, so when that pH is we bring down to pH 1 to 2, then there will be some precipitation, and those precipitations will be of humic acids, and that we can again segregate through centrifugation, right? And the non precipitated portion, that is the fulvic acid fractions. Okay, so now we have humic acid and fulvic acid separated. Now in this humic acid, if we now there are several other fractions we can quantify from this humic and fulvic acid fractions. Means if we extract with alcohol, means we can extract a portion of humic acid through alcohol, that we term as hematomellanic acid. Then again we can redissolve that humic acid in a base and electrolytic solution, means we add some salts. Then some will be precipitated, that is gray humic acid, and then some will be in solution, that is brown humic acid. Means this is in basic solution. Obviously in alkaline solution, the humic acid is basically soluble. So when that soluble thing is there, and if we add some electrolyte, means increase the concentration, then whatever is in the solution, that is known as brown humic acid, and which is precipitated, means not soluble, that is gray humic acid. But then again, so that is all about extraction. Now comes the purification of this humic acids. So for purification purpose, what we do, we take this humic acid, and first we need to remove the let us first talk about humic acid, because whatever humic acid we are getting, that is in terms of solid material. So that humic acid, what impurities it may contain. It may contain some clay portions, some clay particles, some soil particles, basically mostly silicate structure it can contain. So that silicate structure we need to first dissolve. For that, we treat it with hydrogen fluoride, means hydrofluoric acid, and hydrochloric acid, HFCl mixture. So that will dissolve all those silicate materials, and it will be free from clay. So that is the purification of humic acid. Now again, then there is another impurity, that is mostly the different ions that we only introduce in the system, like HF, HCl, or several other elements. So then again, we need to remove those ions, and that we can do by through a electrodialysis, means through dialysis in water. Okay, so that then we can remove those things, and we can have a pure form of this humic acid. And similarly for fulvic acid, it can again, we can pass through an ion exchange regime, and we can elute the pure fulvic acid, means hydrogen saturated fulvic acid. So that is the purification step, and I have actually given you the step-by-step process here. Soil plus this alkali extractant solution, 0.5 normal NaOH plus 0.1 normal sodium pyrophosphate. Then it can be extracted by the same process that I was just discussing, and means it contains about 16 hours shaking in a non-reactive nitrogen atmosphere, and then separation of the dark supernatant, which is basically humic acid plus fulvic acid. Then we reduce the pH to 1 to 2, and then whatever is solid, that is humic acid, whatever is soluble, that is fulvic acid. And then again we purify this humic acid with 0.1 molar HCl and 0.3 molar hydrofluoric acid, shake it in centrifuge tube, shake overnight, and then we repeat this treatment for 2 to 3 times to remove all the ashes, all the silicate structure, all the silicate or hybrid structures maybe. And then again after that we need to go for dialysis. So then we transfer those precipitates of humic acid into whisking dialysis tubes, and then we dialyze against distilled water until the dialysis water gives a negative chlorine test. So if it's free from chlorine, then we say it's free from all the excess ions. So now we have our humic acid, very pure form. Then we can small information about how to characterize those organic matter. Now we have our very pure humic acid particles, which we have separated from our soil. Now we can now this humic acid, this may have different amount of structural groups, different types of structural groups, maybe amino groups, amine groups, quinone groups, carboxylic groups, ester groups, ether groups. So that actually say a lot about the type of those humic acids. it थोड़ा सा ना धीरे करना, ठीक है? So that actually tells us a lot about its structure, how that humic acid is formed, and mostly the condition of that field. Means obviously the effect of some treatments. So that we can say the quality of that carbon we can tell, and just by studying the humic acid structure, characterizing those humic acids. So a easy and very good way of studying the humic acid characteristics is infrared spectroscopy. Now, in this case, obviously we know means the infrared is passed through this humic acid, and this is basically a IR spectrometer. Here we take soil in this, make small tablets, and means in powder form. Means this your humic acid we take in powder form, and we pass the infrared radiation through this sample, and due to this one, now every chemical bond in the structure of the humic acid, they have a certain frequency, and means they stretch and vibrate according to certain frequency. Now when the IR light of same frequency as any of the characteristic vibrations falls on the sample, then energy is absorbed and the amplitude of that vibration is increased. Okay, so means every bond actually vibrates in a frequency, and when our IR light falls and when it is of the same frequency, then that portion is enlarged, means amplification is there, and that we see as a peak, right? So and obviously there is two kind of vibrations. One is stretching vibration and one is bending vibration. So these two here we can say symmetric and asymmetric. This is stretching vibration, means there are two these bonds actually it increases, it decreases, it increases again, and it decreases. So obviously that requires that is very characteristics of some bonds having some high energy signature. And then again there is bending vibration. In bending vibration it requires low energy. That means in the low energy IR will be absorbed in this case. Okay, so bending means the bond actually rotates in its means on its axis, as simple as that. So there are 4 types of bending vibrations: scissoring, rocking, wagging, and twisting. So based on that, now, so that is basically the principle of IR spectroscopy. So if we pass IR through this humic acid sample, then we may have this is the FDR spectra of humic acid that we have done in our lab only. So this is the kind of graph we will have. see there are a lot of peaks. Means in case of peaks, that this is basically transmitters, not absorbent. So we'll basically not have peaks. We have the we can say droughts here. That means IR is passed through and it is absorbed by that bond, the different bonds in this position. And this is basically wave number in centimeter inverse. So this is now this one here, this very peak here. This is due to OH stretching of alcoholic and phenolic groups. This here, this corresponds to CH stretching of alkyl groups. And this point here, this peak, this actually corresponds to the CC and CO stretching of aromatic conjugated ketones and quinones, this here. And this peak here, this actually is corresponding to CO stretching of ketone and carboxyl groups. So what we can say from there, that means our humic acid is having this many amount of groups, right? So if our humic acid is having a high amount of this ketone and carboxylic groups, then we can say that our humic acid is much oxidized, right? If it is having a higher amount of conjugated ketone structure or maybe quinone structure. Now this quinone is a structure that is formed in the later stage of humic acid formation. Means after much polymerization, much complexation of the structure, this quinone forms. So if we have a higher amount of quinone in our humic acid, that means it is a very mature humic acid structure, right? And if alkyl means more aromatic structure is present there. If maximum amount of alkyl structure is there, then we also can say this humic acid is of lesser age. It is not very mature humic acid, and it can supply more nutrient to our soils. So these kind of means this kind of things we can say about the humic acid just by studying its FTIR spectra. And then we can also take out different ratios from this this spectra. Means the ratio of peak depth means the ratio of peak depth of this one 1655 to this one, means this quinone divided by alcoholic phenolic, the alcoholic or phenolic groups. This actually is an indicator of condensation and humification. Means more the quinone structure, more the maturity. So it denotes the maturity, and it is known as the humification index, we can say. Then obviously if we can take the ratio of this one here, means 16, this quinone with this alkyl group. This is an indicator of aromaticity. Means more amount of this group is present as compared to the alkyl groups. Then again, the ratio of peak depth of this keto and carboxylic groups along means with this OH stretching, that tells a lot about the redox status of that humic acid. So those things we can say. And then another one index is we know that is E4E6 ratio. So it has been widely used for studying the humic acid fractions, and this is considered to be the inversely related to the degree of condensation and aromaticity of the humic acid substances, and obviously to the degree of humification. Now for determination, what we do, we take the purified humic acid and dissolve it in an alkali in 0.05 normal sodium bicarbonate and measure the absorbance at 465 nanometer and 665 nanometer, and we take the ratio, E4E6 ratio. Now the ratio of humic acids are generally less than 5, and for fulvic acid it ranges from 6 to 9. Okay, so that was about soil organic matter and its characterization. And then now those are basically some chemical fractions of soil organic matter. Let's talk about now. There is another type of stabilization that is more important than this. Means biochemical or chemical fraction, that is the physical means we can say physical forces of this soil organic matter stabilization, and that is mostly we can have through soil aggregation. Means in soil, that we all know, how soil forms. Soil particles are there. They form, means they coalesce with each other, and we have our aggregates. Now, to form those aggregates, what is the cementing material? But one of the important cementing material is obviously organic matter, right? And that organic matter is protected inside that soil aggregate. So when we talk about soil organic carbon sequestration, so we need to know about how much is aggregate protected or physically protected soil organic carbon. That denotes a lot about the stability of that soil organic matter and the carbon sequestration potential of that particular soil. So for that, we actually do a soil aggregate separation through this process. And this is our wet sieve shaker. So here what we do, we basically take three types of sieves. Okay. You can see this is here 2000 micron sieve. Then it's a 1000 micron, means 1 millimeter sieve, 2 millimeter sieve, and then it is 250 micron sieves. Actually, we use another one. It is not seen in that picture. That is 53 micron. Okay. So we put that four sieves one after one atop another, means first two, then one, then 250, and then 53, and then put this total assembly in this wet sieve shaker that you see here. And then we run this, means we take our soils, undisturbed soil, put it obviously after taking that undisturbed soil, we need to make sure that all that is not containing very large amount of some stones or something like that. Okay, so for that you can always sieve that soil, that undisturbed soil, through a 8 mm sieve. So whatever larger stones are there will be there, so those will be already separated. You take the rest amount and you put that soil on that assembly on that 2 mm sieve. Then you can do some and put and then you put this thing in this wet sieve shaker, and have your water here, because here we are doing wet sieving technique. Means we are measuring the stability of aggregates against the erosion forces of water, eroding forces of water. So then first you maintain the water level to the base of this sieve. Okay, so that means we are promoting some capillary rise of water. Means what will be, whatever the pores, micropores will be there in that undisturbed soil, those will be already filled with water. Okay, so after that all the pores are filled with water, then you go for this wet sieve shaking. Means it will be a vertical movement, say for 10 minutes, and in that 10 minutes what will happen, it will be vigorously pushed, means like back and forth, up and down, up and down. So the soil will be facing some eroding forces, and then whatever the aggregates, means they will be breaking down and coming onto the below sieves. So after this 10 minutes, what we will have, we will be having some soil on this, some on every sieves. Now the more than 2000, means basically more than 250 micron, those particles are known as macro aggregates. Okay, and we have used two sieves on that range. So whatever particles will be on that more than 2000 micron 2 millimeter sieve, those are known as large macro aggregates. And the soils present on that 1 millimeter sieve, that is small macro aggregates. And 250 micron to that one, that 1 mm sieve, So that is basically your small macro aggregates. And then the 53 to 250 micron, that is your known as micro aggregates. Okay, so now we have, and whatever portions is coming in less than 53 micron, that is basically your silt particles or clay particles, because less than 53 there will be no aggregation obviously. So these are the basically some fractions we can get through weight sieving. Okay, so obviously we can always means, couple that like large macro and small macro. So we have our macro aggregates, micro aggregates, and we have our silt and clay portions. So then each of these portions, they are having some soil, along with it some organic matter is there. And then what can another thing it can have some light fractions of organic matter. Light fractions means some remnants of root, mostly some remnants of root. So those are not actually protected by soil aggregates, but due to your higher size, it comes in that size range. But this is not actually protected by aggregate, so we need to get rid of that. Now, to do that, we take subsamples from every this size range. We put it in a solution of sodium polytungstate, 1.89 gram per cc. So whatever portions which is heavier than this 1.89, those will fall down, and that will be our aggregates, which is having obviously our particulate organic matter, means our soil organic matter. And whatever will be floating on the sample, that is known as the free organic matter, or we can have those as the light fraction of this. Means this is light fraction that we see here. Our organic matter means those are not actually protected through aggregation. Then again, we take this one. Now, now we have separated that one, that light fraction, which is not aggregate protected. Now, one more impurity, means we cannot impurity. One more thing we may have on the size fractions that we need to avoid for our better understanding, that is the sand. Because we know what is the size of sand particles. Size of sand particles is obviously more than 53 micron. So in each of that size fractions, some sand particles will be present. And as we know, sand particles are kind of a inert particle. It generally do not bind with your this organic matter. So we should report our means aggregate. When we talk about aggregate associated carbon, we should not consider the sand portions, because that will be underestimation then, right? So how we report? We report sand-free soil organic carbon associated with each aggregate fractions. Right? So whatever this, whatever then this aggregates we have after this heavy fraction, then we disperse this thing with sodium hexametaphosphate. Okay, for each of that size fraction, means each of the size fraction. Now, if we add hexametaphosphate, sodium hexametaphosphate with that aggregate, so it will break the aggregation structure chemically, because we are giving it sodium. Sodium is a dispersing agent, not a flocculating agent. So it will then all the aggregate particles, their sand, silt, clay, those will be separated, right? And then we pass that through a 53 micron sieve. Okay, so whatever will be present on that sieve, that will be mostly your sand particle and the large particulate organic matter particles. So okay, and what will be passed through that sieve, though that will be silt and clay, because that is having a lower size range. So now whatever is present in that I put means on that sieve, that thing again we resuspend in a sodium polytoxide solution having density of 1.85 gram per cc, and then because sand is having more density than 1.85, so whatever portions are precipitated in that assembly, that is sand, and whatever is present on floating in that assembly, that is our particulate organic matter. So this portion actually we need to report for each aggregate size fraction. That is our that is not free organic matter, that is not associated with sand particle. This is the amount of aggregate associated or aggregate protected soil organic matter that we generally report. Okay, so that is the thing I wanted to share with you. And again, another just point is there. Now, when we see when this aggregation, if we go a bit deeper in this aggregation of soil, then obviously there will be micro aggregates, then there will be macro aggregates. Now, this each of these aggregates has a life cycle of its own. Say there is some macro aggregate present in soil, obviously. Then after some time, due to some tillage operations or anything, it will be broken down, right? Now broken down, it will be broken down into several micro aggregates, and the organic matter which will be combining that micro aggregates means combining different micro aggregates and forming a macro aggregate, that will be lost. Okay, then again some another means those individual micro aggregates, then again at after some time it can again uh, react, or it can attach itself with some fresh organic matter input and again form another macro aggregate. Okay, so you see, first breakage happens with that macro aggregate, and the organic matter that is associated with that macro aggregate but not associated with micro aggregate, that is not very stable. Okay, now then there is another type of aggregate that is actually present inside those micro aggregate. Now first in that macro aggregate life cycle, the organic matter which is lost, that is inside the macro aggregate but in between the micro aggregates. Then there is some amount of organic matter that is present inside that micro aggregate. So means that is basically combining several soil separates and forming the micro aggregates. So that we can say that is very stable, because in both that aggregate cycles, that organic matter is there safe inside that micro aggregate structure. Now if we consider, say for a particular instance, for say for this very instance, a soil, it is having it can have three types of aggregate protected carbon. One will be first it can have two types of, let's say for argument, two types of organic matter associated, means two types of aggregate associated organic matter. First, it will be having some micro aggregates, individual micro aggregates present in the soil, and uh, organic matter associated with those micro aggregates. Okay, that will be coming in that lower 53 to 250 micron size range. Then there will be organic matter which is associated with macro aggregate itself. Now there is two types. One is associated with macro aggregate but outside the micro aggregate in that macro aggregate structure, and there will be some of the organic matter which is associated inside the micro aggregate and which is again inside the macro aggregate. Okay, so that is the most stable form of aggregate associated carbon, means carbon associated with micro aggregates inside the macro aggregates. So that thing, that aggregate size fraction also we can determine, means through this process here. So here what we do, first we, obviously the first step is similar. So after that we take this macro aggregate here, the portions of macro aggregate, and we take a subsample and we put some glass beads in those subsample, and again we run the this aggregate separation technique, this weight saving technique, and then again we pass it through 53 micron. So then whatever portion is broken through that means glass beads and shaking, and whatever portion is not broken, means stable, that we term as the portion, the micro aggregate, which is associated with the macro aggregates. So that, and that, then again in that portions also we have to go for removal of this light fractions and also removal of the sands, and we can have every aggregate associated or aggregate protected soil organic matter, which is very easy, which is very important for a for seeing the stability of organic matter. So I think that was it. Thank you. So anything, anyone, any clarification, any questions, anyone if you want to ask?
एक्चुअली सर जब हम एनालिसिस करते हैं तो बहुत सारे हमारे पास बुक्स हैं, तो हम किस बुक को रेफर करें एनालिसिस के? आप एक मतलब स्टैंडर्ड को बताइए जिसको रेफर कर।
Actually I should have included that in my slide only. Anyway, तो अभी देखो ना, ये जो इंडियन इंस्टीट्यूट ऑफ इंडियन सोसाइटी ऑफ सॉइल साइंस, it has brought out a book on soil analysis. Okay, so I have that with me. Yes. Actually this one you can have, you can see soil analysis book by Indian Society of Soil Science.
Yes sir, seen sir, seen.
Okay, yeah, so this is, yes. I think in today's today's time, this is the best manual you can follow. Okay, then there is a manual which is also developed in IRI only, SP Datta and co-workers. Then there is one manual developed by Dhyan Singh and his co-workers, Dhyan Singh, Divedi, Chonkar, and then there is Datta, Meena, Divedi, and I guess Shukla. So these are very good manual for daily basis live estimations, so you can follow. But in my opinion, this soil analysis book is best for everything, this routine analysis and also for your JRF preparation. Okay, and if you need to see, if you need to study this process or follow this process for your MSc PhD research, no, then I think for these books are good for following the process. But if you need to know why we do this, what is the use of the reagent, some particular reagent or some time, for that you have to read the Jackson, is the book by ML Jackson. I think 1975 one edition is there, so that is very good one. And then there is another improvement on that book that is also published by this USDA or US by Page. One book is there. That Page book is having your soil mineralogical, physical, chemical, biological, every chapter is there. So you study that one also, to know the principles of this soil different analysis. Okay, so that was all about it. So Suman, all these participants here, they are JRF aspirants or they are in masters or something?
Some of them actually, sir, in there is another work and another program started in the university from 2 days ago, sir. That is the land use planning. So most of the PhD students are involved in these things. So, sir, only JRF and MSc students, SRF candidate are joined this meeting.
ओके, सो मींस यहाँ पे जितने भी लोग हैं सारे मतलब JRF ही देने वाले हैं, राइट? सो प्रिपरेशन शुरू कर दिया सब? मींस if you want to, even any one of the students, if अगर किसी को कुछ पूछना है regarding that preparation, also पूछ सकते हैं। तो इसलिए पढ़ाई तो हो गया सारा, अगर प्रिपरेशन के रिगार्डिंग कुछ पूछना है तो आई थिंक दिस इज द बेस्ट टाइम। ओके, एंड इफ नॉट देन वी कैन जस्ट
एसआरएफ के लिए सर अपने को कौन से स्पेसिस्ट सर एसआरएफ के लिए अपने को सर किस तरह की स्ट्रेटजी अपनाना चाहिए फॉर 2 to 3 months, हाउ टू कवर ऑल दीस थिंग्स इन शॉर्ट मैनर?
देखो एसआरएफ के लिए भी क्या है ना कि पहले तो आपको ना थोड़ा क्या बताए, सी इफ वी टॉक, okay, let us just have another session of 10 minutes of this thing, how to prepare for these exams. See, if we have means पहले बात करते हैं कि प्रिपरेशन स्ट्रैटेजी, ठीक है? तो प्रिपरेशन स्ट्रैटेजी में आपका सिलेबस जो है दैट वो सिलेबस भगवान है वहां पे, ठीक है? सिलेबस से बाहर से कुछ नहीं आएगा जनरली, और सिलेबस में जो जो चीज है वो बहुत ज्यादा इंपॉर्टेंट है। सो फर्स्ट थिंग, यू शुड नॉट ओमिट, मींस वो सिलेबस हमेशा आपके आंखों के सामने होना चाहिए, वो चीज आपको पढ़ना है, कंप्लीट करना है, नंबर 1। नंबर 2, पहले आप देखो कि आपका सिलेबस के अनुसार सबसे बढ़िया बुक कौन सा है। अगर आप मेरे से पूछो आज के डेट पे जो सॉइल साइंस का, फिजिकल साइंस का जो भी है, तो उसमें अगर एनालिसिस का पोर्शन कवर करोगे तो ये इंडियन सोसाइटी ऑफ सॉइल साइंस वाला ये बुक बहुत बढ़िया है। दूसरा, जो तुम जब फर्टिलिटी की बात करते हो तो फर्टिलिटी के मामले में भी जो इंडियन सोसाइटी ऑफ सॉइल साइंस का जो बुक है काफी कॉम्प्रिहेंसिव है वो, ठीक है? देन अगर आप पेडोलॉजी का बात करते हो तो पेडोलॉजी के लिए जेल साइकिल से बेहतर कुछ नहीं होता। देन अगर आप बायोलॉजी में आ जाओ तो बायोलॉजी में भी इंडियन सोसाइटी ऑफ सॉइल साइंस वाला जो बुक है बढ़िया है। उसके साथ एक एस कॉइनी या एल कॉइनी, आई डोंट रिमेम्बर द एग्जैक्ट नेम ऑफ द ऑथर, तो कॉइने का एक बुक आता है COYNE, कॉइनी, मोस्ट प्रोबब्ली एस कॉइनी, तो उसका एक बुक आता है माइक्रोलॉजी का, पतला सा है, दैट एक्चुअली कवर्स अ लॉट और बहुत अच्छे से कवर करता है। तो ये माइक्रोबायोलॉजी हो गया। तो यही है एकदम बेसिक बुक। आपने मेरे को बोल रहा है कि 2-3 महीना, तो मैं इसलिए ये बोल रहा हूँ कि तो ये चीज आपको पहले कवर करनी है, और ये चीज हमेशा देखना है कि जो आपका सिलेबस दिया हुआ है, क्योंकि ये सारे बुक्स में ऑब्वियसली पूरा सिलेबस तो कवर नहीं होगा, फिर भी कुछ कुछ चीज रह जाएगा। तो वो चीज के लिए आपको फिर दूसरा बुक ढूंढना पड़ेगा, कौन से बुक में ये चीज अच्छे से दिया हुआ है, ठीक है? मे बी आप ब्रैडी में ढूंढिए, ब्रैडी तो इज लाइक व्हाट आई कैन से महाभारत ऑफ सॉइल साइंस, उसमें आपको सब कुछ मिल जाता है जनरली। तो वो ब्रैडी में खोज के आपको देखना पड़ेगा कि कहाँ कौन सा चीज है। तो ऐसे स्ट्रैटेजी अपनानी चाहिए फॉर जेआरएफ बोल लो, एसआरएफ बोल लो, अगर ऐसे करेंगे तो अच्छा है। और फॉर दैट मैटर पहले ये बेसिक बुक ही करो, उसके बाद एडवांस्ड अगर टाइम हो तो बाकी एडवांस बुक्स पे जाओ, कि 10 वाला थोड़ा पढ़ लिया, फिर टिसडेल लेंसल का बहुत अच्छा बुक है फॉर फर्टिलिटी पर्सपेक्टिव, उसको थोड़ा सा वहां से देख लिया, ये सेकंड में है। एंड प्रिपरेशन के टाइम पे तो ये तो हो गया पहला स्टेप। अब सेकंड स्टेप में आता है आपका प्रिपरेशन। अगर आपने एमसीक्यू क्वेश्चंस के लिए प्रिपरेशन कर रहे हैं, ठीक है, तो उसमें आपका बुक जो पढ़ना है वो बहुत अच्छे से पढ़ना है, मतलब एकदम लाइन बाय लाइन बहुत अच्छे से। एंड डू नॉट जस्ट मग अप ऑल दिस थिंग्स, मतलब उसको ऐसे मगिंग अप मत करिए, उसको समझिए, समझ के पढ़िए थोड़ा, ठीक है? एंड व्हेनेवर यू आर स्टार्टिंग दिस जर्नी ऑफ प्रिपरेशन फॉर एनी एग्जाम, तो फर्स्ट start with the easier chapters. That is always best for easier. अगर हम soil science की बात करें तो मेरे हिसाब से सबसे easy chapter soil science में soil fertility का आता है. वो बहुत easy होता है, उसमें ज्यादा कुछ नहीं है, छोटा छोटा थोड़ा प्रिंसिपल है. और अगर हम हार्ड बोले तो soil mineralogy बहुत हार्ड है उधर. soil chemistry जो एकदम कोर केमिस्ट्री होता है वो बहुत हार्ड है वहां पे. ठीक है, तो उसको लास्ट में लीजिए. पहले जो इजी पोर्शन है उसी से शुरू करिए. एंड पढ़ाई का जो टाइम है ना, वो टाइम पहले आप 3-4 घंटे अगर दिन में पढ़ सकते हो तो ठीक है. अब धीरे धीरे वो टाइम को इंक्रीज कीजिए. जैसे जैसे आपने आगे बढ़ते हैं अपने इस जर्नी में, वो टाइम को थोड़ा इंक्रीज कीजिए. और जैसे हमने बोला ना कि कोई नया चीज है तो उसको फिर आगे से लिख के या फिर कहीं पे एक नोट बना के वो पेज अपने बुक में डाल लिया, तो ऐसे आपका फिर बनता जाएगा. ठीक है. और ये पूरा प्रिपरेशन में रिवीजन बहुत इंपॉर्टेंट है, क्योंकि आप जब भी कुछ पढ़ोगे ये तो पक्का है कि 10 दिन बाद आप उसको भूल ही जाओगे, इसमें कोई दोहराया नहीं. तो उसको रिवाइज करना पड़ेगा फिर. ठीक है. अब जितना ज्यादा बार आप रिवीजन करोगे उतना ज्यादा अच्छा है. आपने अगर 10 बार रिवीजन किया एक बुक का तो बहुत बढ़िया है, बट एक बार पढ़ के आप जाओगे तो ऑब्वियसली उसमें ना के बराबर ही चांस है. तो बहुत ज्यादा रिवीजन करने की जरूरत है. एंड ये जब प्रिपरेशन हो रहा है, अब इसमें बहुत सारा चीज ऐसा आता है कि जिसमें ना बहुत डेटा होता है जो हमें समझ नहीं आता. मैं पहले ही बोला कि समझ के पढ़ना है, मगअप नहीं करना है. तो बहुत सारा चीज ऐसा है कि जहां पे समझने का कुछ है ही नहीं, वो पूरा मगिंग अप का ही सीन है. जैसे कि कोई स्पेसिंग हो गया या फिर कोई प्रोडक्शन डेटा हो गया. ठीक है, ये सब चीजें. तो उसका आप चार्ट बनाओ. यू मेक सम चार्ट, यू पेस्ट ऑन योर वॉल्स. उसको आपको पढ़ने की जरूरत नहीं है, देखने का अलग से पढ़ने की जरूरत नहीं है. आप हर टाइम जब सोने आएंगे तो वो आपके आंख में आ जाएगा, तो वो ऐसे ही रह जाएगा, उसको अलग से पढ़ने की जरूरत है नहीं. एंड उसके बाद जो इंपॉर्टेंट चीज आता है वो है डिस्कशन विथ योर फेलो पार्टनर्स. तो उसको डिस्कस करिए थोड़ा. कि आपने किसी से मिल लिया तो उसको आपने 4 क्वेश्चन पूछ लिया. आपने सॉइल फर्टिलिटी पढ़ा, उसको जाके पूछ लिया कि भाई बताओ सॉइल जो एसिड वाले सॉइल में फास्फोरस हम कैसे करेंगे. ऐसे उसने आपको 2 क्वेश्चन पूछ लिया. ये चीज बहुत फायदेमंद है चीजें याद रखने के लिए. ठीक है. देन एग्जाम दीजिए, मॉक एग्जाम दीजिए, क्योंकि आपने एग्जाम हॉल में जाके बहुत सारी गलती करनी है, उससे आपका रैंक पीछे जाता है. तो वो गलती आपने पहले कर लीजिए अपने मॉक एग्जाम के टाइम पे. तो इसलिए एग्जाम, मॉक एग्जाम्स बहुत इंपॉर्टेंट है और उसको टाइम देख के देना है. मतलब अगर आधे घंटे का पेपर आपने बनाया, आप अपने फ्रेंड के लिए ये बनाइए, आपके फ्रेंड के लिए, फ्रेंड को बोलिए आपके लिए एक पेपर सेट करे, या फिर जो पुराने जेआरएफ के पेपर्स है सारे बुक्स में दिए हुए होते हैं बहुत सारे ये कॉम्पिटिटिव वाले बुक्स में, उसको सॉल्व कीजिए. मतलब उसको आंसर मत पढ़िए, उसको सॉल्व कीजिए, टेकिंग केयर ऑफ द टाइम लिमिट. सो आपका बहुत इजी हो जाएगा उसमें. सो ये एमसीक्यू एग्जाम के लिए ये सारे पॉइंट तो है ड्यूरिंग प्रिपरेशन. नाउ अगर हम बात करें कि एमसीक्यू एग्जाम देना कैसे है, मतलब इन द एग्जाम हॉल, हाउ विल यू अप्रोच दैट पेपर. नाउ सी, पेपर में आपका क्या है वहां पे कि यू से फॉर एग्जांपल आई डोंट रिमेंबर हाउ मेनी क्वेश्चंस जनरली दिस डेज आर. सो सी से वहां पे 100 क्वेश्चन है, आपके पास 100 मिनट्स है. वैसे आप बताओ अभी क्वेश्चन का पैटर्न क्या होता है. 2 घंटे का टाइम होता है.
120 question is here that time.
120.
Yes.
हेलो, 120 ना.
Yes, yes.
120 questions in 2 hours.
Yes, yes, sir.
Okay, okay. So
Per
that
question 1
means
minute. Sir.
हाँ, पर क्वेश्चन 1 मिनट. हाँ ठीक है. तो आपका म्यूट कर लो यार, थोड़ा बहुत आवाज आ रहा है वहां से फैन का. तो पर क्वेश्चन ऑब्वियसली इट इज 1 मिनट. तो अब ये 1 मिनट वाला सीन यहाँ पे वैसे है नहीं. इसको आपको पहले क्या करना है, ये तो एमसीक्यू है एसआरएफ में, ऑब्वियसली एमसीक्यू एग्जाम इज देयर। तो आपको पहले क्वेश्चन से ना हर एक क्वेश्चन को इंडिविजुअली करना है, क्वेश्चन स्किप मत कीजिए। तो पहले आपने गया क्वेश्चन को पढ़ा। अब क्वेश्चन पढ़ने का भी एक तरीका होता है। पहला जब क्वेश्चन है उसका जो ऑप्शंस दिए होते हैं उसको आप मत पढ़ो, ठीक है। पहले आपने क्वेश्चन को पढ़ो एंड रीड द क्वेश्चन वेरी क्लोजली, मींस वेरी क्या बोलेंगे उसको, बहुत अच्छे तरीके से पढ़िए, बारीकी से पढ़िए कि व्हाट दैट क्वेश्चन एक्चुअली टेल्स। क्योंकि हमने बहुत कभी कभी बहुत जल्दी के चक्कर में ना क्वेश्चन का, जैसे इट इज हार्मफुल, इट इज नॉट हार्मफुल, इट इज बेसिकली टू ऑपोजिट स्टेटमेंट्स, तो ऐसा नहीं होना चाहिए। इसलिए वो क्वेश्चन को बहुत अच्छे से पढ़ना चाहिए। And whenever you are reading that question, do not see the answers. That this is very important. जब आपने क्वेश्चन पढ़ेगा वहां पे कुछ लिखा हुआ होगा, सो वो फिर आपको ना सीधा ले जाता है, मतलब देन you think the answer in your head, maybe in some minutes of like 2, 3, 4 seconds maybe. So उसमें आपके दिमाग में कुछ ना कुछ आंसर आएगा, कुछ इमेज आएगा। हम जब भी पढ़ते आवर मेमोरी आर स्टोर्ड इन इमेजेस, सो कहीं ना कहीं से कोई ना कोई इमेज, कोई कुछ किसी बुक का कोई इमेज आपके जहन में तब आएगा। देन यू सी द आंसर। उसके तब क्या होगा, कौन सा आंसर उसके लिए एकदम फिट है आपको पता चल जाएगा। आपने उसको मार दो, टिक कर दो, ऑप्शन को टिक कर दो। तो ये आपका एक क्वेश्चन हो गया। ऐसे ही दूसरा क्वेश्चन, ऐसे ही तीसरा क्वेश्चन, ऐसे चौथा पांचवा करके एकदम लास्ट तक जाना है 120 नंबर तक, ठीक है। इसमें कोई मतलब और कोई कुछ मत करिएगा, ये एक एक करके हर एक क्वेश्चन पे इंडिविजुअली ऐसा टाइम डाल के आपको करना है 120 तक। अब क्या होगा, इस प्रोसेस से अगर करेंगे तो मेरे हिसाब से बहुत सारा क्वेश्चन जो आपने एक्चुअली पढ़ा हुआ है वो आपका हो जाएगा, ज्यादा उसमें दिक्कत नहीं होगा। अब उसके बाद भी कुछ कुछ क्वेश्चन ऐसे होंगे जो थोड़ा सा क्रिटिकल है, मतलब आपको उसका आंसर एग्जैक्टली नहीं पता। तो आपने ऑलरेडी उस क्वेश्चन को पढ़ा, उसको सोचा, फिर आपने आंसर, आपको कुछ दिमाग में थोड़ा बहुत कुछ इमेज आया, उसके साथ आपने आंसर देखा, जो मैच हुआ आपने टिक कर दिया। अब कहीं पे जाके आपको कंफ्यूजन होगा कि इसमें से कौन सा आंसर है। आपके पास 4 ऑप्शन है, अब इसमें से मेरे को 2 में कंफ्यूजन है। तो फर्स्ट अटेम्प्ट में वो कंफ्यूजन क्लियर करने के लिए मत बैठिए। पहला आपने सोच लिया, सोचने के बाद जो दिमाग में आया उसको मार के निकल गए। वहां पे अगर कंफ्यूजन हुआ तो उसको मत करिए, कुछ करिए, उसको छोड़ दीजिए, ठीक है। अब ये फर्स्ट अटेम्प्ट जो है, आपका जो टोटल अलॉटेड टाइम है उसका 60% टाइम में इसको कर लीजिए। मे बी आपका 2 घंटा टाइम है ना टोटल, तो आपने उसको 1 घंटा 10 मिनट में भी, 1 घंटा 10-15 मिनट में आपको हो जाना चाहिए ये फर्स्ट अटेम्प्ट। तो उसके बाद आपके पास ऑलरेडी है कि आपने कौन सा क्वेश्चन छूटा हुआ है, फिर उसको लेके बैठिए। मगर अब यहाँ पे भी एक चीज इंपॉर्टेंट है कि जो चीज आपने पहले फर्स्ट अटेम्प्ट में कंप्लीट कर लिया उसको वापस घूम के नहीं देखना है। वापस अगर घूम के देखोगे, उस पर ज्यादा सोचोगे तो गलत करोगे। ये आप मेरे से लिख लो, आई एम क्वाइट श्योर अबाउट दैट। नेक्स्ट टाइम उस पर आप जाओगे, जो आंसर मार्क करोगे उसको वो गलत ही होगा। क्योंकि आपका दिमाग जब सोचता है ना, फर्स्ट अटेम्प्ट में वो जो सोचता है, जहां पे इंडिकेट करता है वही सही होता है, क्योंकि वो अनबायस्ड है। आपने जैसे ऑप्शंस देखा उसके बाद आपने फिर सोचने गए तो एक बायसनेस आ गया आपके में, तो आप उसको फिर गलत ही करना है आपको। तो ऐसे फिर वो सेकंड वाला जो आपका कंफ्यूजन वाला क्वेश्चन है, अभी उसको लेके आप वन बाय वन करिए। अब उसमें भी अगर आप थोड़ा सोचेंगे, अब ये सेकंड वाला जो मतलब ये रिवीजन का टाइम है ना क्वेश्चन पेपर को, तो उसमें आपको ऑब्वियसली वही सब क्वेश्चन है जिसमें थोड़ा ज्यादा सोचना है। एंड ऑल्सो द क्वेश्चंस जहां पे ना थोड़ा मैथमेटिकल कुछ प्रॉब्लम्स है, लाइक सॉइल बल्क डेंसिटी, मॉइस्चर कंटेंट, एटसेट्रा एटसेट्रा। तो ऑब्वियसली वो सब चीज तो 5 सेकंड में नहीं आएगा, उसके लिए आपको कैलकुलेशन करना पड़ेगा। तो वो सब चीजों को भी इसी सेकंड अटेम्प्ट में रखिए। तो एक बार फिर से उस क्वेश्चन को सब में फिर थोड़ा पेन लेके सोचिए, सोच के देन यू टिक द आंसर्स. And even if that second attempt also you are finding some questions those you have not heard actually, मतलब आपको पता नहीं है कुछ भी, वो नाम बस सुना हुआ है बट उसके अंदर आपको कुछ नहीं पता, उसको फिर उस अटेम्प्ट में भी छोड़ दीजिए. तो ऐसे ये रिवीजन के बाद भी आपका ना लास्ट में फिर बचेगा लाइक 10-15 कुछ क्वेश्चन बच जाएंगे जो आपने अभी टच नहीं किया. अब लास्ट में उसको लेके बैठिए और उसको फिर थोड़ा ना ये डिडक्शन लगाइए उसमें. सपोज यू डोंट एग्जैक्टली नो द आंसर बट यू नो सम अदर थिंग्स. जैसे फॉर एग्जाम्पल जस्ट मेरे दिमाग में जो आ रहा है, से कोई ऐसा क्वेश्चन है जिसमें मान लो पूछा गया है से कोई एक प्रोग्राम है, ये प्रोग्राम कब आया था, एक्सटेंशन का है वैसे थॉउजेंड्स का है नहीं, कि ऐसा एक प्रोग्राम है ये इंडिया गवर्नमेंट ने कब लॉन्च किया. अब आपको एग्जैक्टली ये पता नहीं है कि इसका एग्जैक्ट ईयर कौन सा है, बट आपको ये पता है कि ये ना वो जब वो बांग्लादेश का वार हुआ था ना, वो बांग्लादेश लिबरेशन, उस टाइम पे ना ये आया हुआ था. तो ऑब्वियसली ये 1971 के आसपास होगा. तो फिर जो आंसर आपको वहां पे लगे तो उसको टिक मार देना है. ऐसे करके करो. और एमसीक्यू क्वेश्चन के लिए ऑलवेज ट्राई फॉर कि आपने ना 100% वहां पे करके आना है. क्योंकि आप देखो कि आपका अगर आप उसमें भी जाओगे ना कि 4 आपका क्वेश्चन करोगे तो उसमें क्या एक क्वेश्चन अगर सही हो गया और 3 गलत हो गया तो भी तो आपका तो कोई प्रॉब्लम नहीं है, सेम सेम है. मगर उसमें अगर आपका 2 क्वेश्चन सही हो गया तो आप तो बेनिफिट में रहे ना. तो ये थोड़ा सा आपको कैलकुलेटेड गेस वहां पे लगाना है. और ये कैलकुलेटेड गेस पहले से नहीं लगाना है. पहले जो श्योर शॉट जो पहले 1 घंटा 10 मिनट का खेल बोला वो है कि आपका एकदम जो आपने पढ़ के गया आपको सब पता है, वो क्वेश्चन गलत नहीं होने चाहिए, वो आपका मार्क्स है. और उसके बाद सेकंड रिवीजन थर्ड रिवीजन में आपका कैलकुलेटेड गेस है, करके मैक्सिमम करके आओ बस. दैट्स इट. सो एनीवे यही सब है और क्या. थैंक यू फॉर इनवाइटिंग मी. सो मे आई लीव.
आपने आज शॉर्ट नोटिस पे टाइम देकर इसको पूरा किया सर, इसके लिए थैंक यू सर.
हाँ थैंक यू.
और सबको मेरे
ठीक है.
ख्याल से और लास्ट में आपने ये भी मतलब कि अपने को तैयारी कैसे करनी है. एवरीवन विल बी बेनिफिटेड फॉर ऑल दीस थिंग्स.
हाँ ये ठीक है, ऐसा ही करो. ये अपने एक्सपीरियंस है और कुछ नहीं. हमने भी कभी आपके तरह एग्जाम हम भी लिखे हैं तो वही चीज. ठीक है, बेस्ट ऑफ लक. ठीक है फिर. ओके थैंक यू.
थैंक यू सर. थैंक
यू. थैंक यू.
यू. आई